why the deoxygenated blood is not as red, or when we draw a diagram, why do we give a blue color? Just to but there is scientific basis for that. So, what is the basis? Carbaminohemoglobin has a distinctive blue color. Hello, what are we talking about? Hemoglobin. It has been in the tissues, it has picked up carbon dioxide, and what color it has gained now? Blue color. Therefore, contribute to the dark blue color, that may contribute to the dark red color of the deoxygenated venous blood. That blue color, whatever is the color of the venous blood, that bluish tinge there is because it is the carbaminohemoglobin. Now, follow the story. We are still talking about carbon dioxide being transported by the blood. Now, some of the facts. Take the blood 100 ml, which has been fully oxygenated at the level of lungs and returning to the heart. So far, and that blood will enter into the left auricle, go to the left ventricle, and then it will be pumped on the aorta. Let us presume that we are collecting the blood from aorta. How much of blood have we collected? 100 ml. And I will try to find out how much of carbon dioxide is there in that, and I find that it is about 48 ml. All put together, whatever it is how much is there? 48 ml. Are you so far? So that blood which is and and that same 100 ml, how much of oxygen is present in that? 20 ml. How much is there? He is right. How much is there? 20 ml. So that is what in terms of actual volume. My 100 ml of that blood is going to contain 48 ml of what? And what ml of oxygen? 20. Now, that blood goes into the tissue, and Bohr effects, etc., whatever happens, and then picks up the oxygen. Now, I am going to collect the blood from the venous system. Obviously, it is going to have more carbon dioxide, and that carbon dioxide I find is about 52 ml. How much it is? So, actually how much was added? 4 ml was added. Now, I will take this blood from 52 back to the lungs, and there I will get rid of carbon dioxide. How much of carbon dioxide I am getting rid of? Again 4, again 4. So, it oscillates between 48, it does not go below 48 and does not go above 52. If you are healthy, and if you are breathing normally and oxygen is abundantly available, 48, 52, 48, 52. Just as oxygen was 20 in the lungs, 15 in the tissue, you are in that range. Of course, there is some margin, but you are in that range. Are you so far? Good, good. So, where are we? We are this is the we had oxygen dissociation curve, yes or no? So, we are here we have a carbon dioxide dissociation curve. Of course, it is going to pick up carbon dioxide in the tissues and give it up in the lungs, and we have seen how much? 4 ml every time, 4 ml per 100 ml of blood. Good, good. Now, let us talk purely in terms of partial pressure. Your oxygen, blood coming from the lungs, 100 ml contains how much of carbon dioxide I told you just now? 48 ml, 48 ml. In terms of partial pressure, how much it is? 40. How much? Very good. How much it is? 40. So, partial pressure of 40 means 48 ml of CO2. Hello, are you okay so far? We are all good so far. Then you take this blood to the tissues, and when you go to the tissues, you pick up carbon dioxide. So, you go from, in terms of partial pressure, you go from 40 to 45, but in terms of volume, you go from 48 to 52. No, not Greek and Latin, very simple. I am talking the same thing in two different languages. In the language of partial pressure, I am going from, in the tissue, I am going from 40 to 45 partial pressure. In terms of actual volume, I am going from where to where? 48 to 52. Now, you would appreciate this. Where are we? Where are we going? What does this indicate? Please, on the x axis, what do we have? 40 to 45, just see there, you appreciate that 40 to 45 there. Okay. And from there if you go to the x axis, what do you get? 48 to 52. What do you get? You see, from if I take these 2 points here on this line, and if I go here, what do I get? Okay. So, this is the these are the two extremes in which the carbon dioxide is operating as we go from the arterial blood to the vein and back to that. Okay. Now, life is more interesting than that. Just as there we had Bohr effect, where carbon dioxide was influencing the affinity of oxygen to hemoglobin. It was different in the lungs, different in the tissues. Okay. Reverse is also true. Means what? Means what? Means what? Oxygen is influencing. Well, remember, you remember 23% of carbon dioxide is carried in the blood in the form of what? Carbaminohemoglobin. So, the hemoglobin molecule, as it goes, which is sitting in the RBC, and as it is going from the tissues, okay, it has picked up carbon dioxide, which is 23% of the total, okay, and that 23% of that carbon dioxide molecule is combining with hemoglobin. Okay. That act itself is influencing the affinity of hemoglobin to oxygen. In what direction? I think I am absolutely sure you can guess. The more the carbaminohemoglobin is formed as a result of carbon dioxide being picked up by the tissues, okay, it reduces the affinity for oxygen. So, you give more oxygen. One more amazing trick played by the hemoglobin molecule. Okay, so, the moment you become carbaminohemoglobin, you give more oxygen, okay, and opposite will happen at the level of lungs, okay, where the moment carbaminohemoglobin gives away carbon or gives away carbon dioxide, which was bound to hemoglobin, okay, its affinity for oxygen will go up. This is called as Haldane effect. What is it called as? Say that again. This is called as what? Haldane effect. Okay. So, now let us see, this is the, so actually for this, in the case of, it is very interesting to understand this figure, I will try to explain it to you. Let us just consider the very simple, the blood is coming from the lungs and going to the tissues, good. Partial pressure of carbon dioxide is 40 mmHg. Okay. So, partial pressure is 40 mmHg. And how much of how much is the carbon dioxide? 48. We have done that. Okay. All right. Now, the blood has gone to the tissues. In the tissues, it has picked up carbon dioxide. Okay. It has gone, the partial pressure of carbon dioxide in the blood has gone from 40 to 45. Okay. Now, when that is happening, when the carbon dioxide has gone from 40 to 45, the blood has lost oxygen. Hello, we are in the tissues. The blood has what? It has lost oxygen. Okay. As a result of that, the hemoglobin molecule, okay, has more affinity for, it can combine with more carbon dioxide. As a result of that, or because it combines, the curve shifts in such a way that when you are talking about the partial pressure of oxygen is 40 mmHg, that is in the tissues, okay, you have the curve here where now you can carry 52 ml of oxygen. If that were not to happen, now to appreciate the point, if that were not to happen and if this were the only curve, then this will go from here to here and here to here, means the blood can 100 ml of blood can go from 48 to 50, which means increase in what? 2 ml. But because oxygen has gone away, hemoglobin can combine more readily with carbon dioxide. Therefore, the same hemoglobin now can carry 4 ml of carbon dioxide, exactly opposite of Bohr effect. And physiologists tell us that in practice Haldane effect is even more useful than the Bohr effect, and it is more profound. So, please remember, please try to now I am going to tell you a who is he? Read for me, John Scott Haldane. Okay, he came up with, of course, that is him. Now, another trivia about Haldane. He had a son. The son's name was John Burden Sanderson Haldane, Fellow of Royal Society. Okay. Now, why am I talking about his son?
He is the one who gave the primordial soup idea, right?
Very much so. In 1956 or so, he had some, this is not science, this is trivia in science, which makes science more interesting. In 1956, he had a problem with British government. Who? The son. Okay? And he said, goodbye, I do not like you. And then what did he do? He came to India. Where did he go? Bhubaneswar. He worked in Bhubaneswar, okay? And there he died. So, his grave is in Bhubaneswar. Okay? So, Haldane is Indians take that name Haldane with a lot of with a fondness, okay? Because he took Indian citizenship. Who? Not the dad, okay? Okay, what is it's complicated? John Burden, whatever it is, okay? And he was, look at his. I want you the British Indian scientist, okay? Can you read there? And what about him? Known for his work in the study of physiology, genetics, evolutionary biology, and mathematics. He made innovative contributions in the field of statistics and biostatistics. He was the son of, okay. So, this is just an interesting side story I thought we should share, and particularly known about Haldane because this name will come very often in several, okay. Now, we are hemoglobin molecule is still not done with the tricks, still more tricks, amazing. A molecule never ceases to amaze you, beautiful molecule. In the RBC, there is yet another very small compound called as 2,3-diphosphoglycerate. Please pronounce it for me once. Abbreviated as what? 2,3-DPG. It is a very small molecule. Okay. Now, we are looking at this is a cartoon. This is a cartoon of hemoglobin molecule, and I am sure you can appreciate the 4 subunits are there. Each subunit is bound to an oxygen. And more or less author tells us that this is somehow the orientation in which the 4 units are associated with one another. Now, you take this molecule, this is obviously all 4 oxygen. Can you appreciate here O2, 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 it is a saturated molecule, and obviously this is in the lungs and this goes into the tissues, and once it starts going into the tissues, it loses one molecule of oxygen, second, third is gone, but the 4th is not gone yet, it will go. As a result of the losing away or giving away of the oxygen, there is some change in the dimension of one unit with reference to another unit. And then in the gap between the 2 subunits, there is a little more gap, and in that gap fits the molecule. What is that molecule? 2,3-DPG. Okay. And so once that molecule, so 2,3-DPG, whereas it has no affinity for the fully oxygenated molecule, it has a great affinity for the molecule which is partly deoxygenated. And once it sits, goes and occupies in that, can you see there the 2,3-DPG is sitting there in the second one? When it sits, that last oxygen which was sitting there is readily given away. So this is yet another trick how the cell ensures that when you are in the tissue, Okay. You want to make sure that you give as much as oxygen as possible, and for that it has this molecule which is it has. So, is this molecule there in the so is the 2,3 DPG molecule abundantly present in the RBC? Yeah, it is. Not only that, it has been found that when you do exercise, it goes up. Okay. Okay. Look at the beauty of physiology. Okay. Because when you are if you have been doing exercise, obviously your requirement for oxygen has gone up and your physiology responds. How? By generating more DPG, so that you can give away the oxygen molecule. So, this is the structure of DPG. Please more.
DPG is coming from where? Blood?
It is in the blood. It is in the RBC. DPG is abundantly present. Of course, it has to interact with hemoglobin. No? It has to interact with hemoglobin. It has to be in the RBC. It is in the RBC. Have no doubt.
But when you are doing the exercise, the DPG level is increasing. So
Though
DPG is coming from the blood or
which one? Which one?
2,3 DPG.
2,3 DPG. We don't really know. We don't really know. That's a very good question. Where is it coming from? Because RBC of course cannot synthesize new DPG. Okay, we don't know. Maybe some literature is there. I have to read. What's your question? Say the frame your question again. I'll read. Let's see. Okay. Where is it? Where and how is it coming from? We'll try to find out. Okay. Now this DPG has a very interesting role. We are looking at a pregnant mother. Okay, and in her uterus you can see the fetus there. Are you okay there? And you can see that red placenta is there. And why placenta is important? Because the baby is now, of course, the baby does not have its own lungs. So, it is dependent on oxygen. So, baby is actually dependent on the lungs of the mother. So, the mother gets the oxygen in the blood, and then the oxygen goes to the through the blood, it goes to the placenta. In the placenta the gaseous exchange will happen. Supposing both the fetus and the hemoglobin of the fetus and hemoglobin of the mother, they are of the they are same. They are not same. Okay, I am going to that. They are same. Okay. So, the depending on the partial pressure, of course, it will go. It will go, but again the beauty of hemoglobin, and I will explain it to you how it works. 4 subunits, 2 alpha, 2 beta. Are you okay? 2 alpha, 2 beta. When the baby is developing in the uterus of the mother, okay, there are different forms. There are different forms of hemoglobin, and they just as so as adults, what do we have? 2 alpha hemoglobin molecule, 2 alpha, 2 beta. Are you with me? 4. Are you okay so far? The baby will have 2 alpha. So, this is the week, this is the point of birth. So, from here to here the baby is developing in the uterus of the mother, here the baby is born, and this is beyond that. Right from the very as the blood is being synthesized, you have alpha, 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 alpha. So, you collect the blood from the baby right from the beginning, you will have hemoglobin has 4 units, 2 units are alpha, done. But other 2 units are not beta, the other 2 units are maybe what is this, I do not know what is this, zeta, and then what is this, epsilon, whatever, and delta, gamma. So, these 2 versions come very early and go, but I will draw your attention to the beta unit is low at the time of the birth, only so much of beta is there. So, if you take the blood of a fetus, you will find that in early stages it contains mainly of course, the 2 units are alpha and 2 units are what? Are you getting it? It is not the same, it is not the same, it is not the same. It is only after the birth or so, much after the birth, after 20 weeks of birth, that you have the adult composition of 2 alpha units and 2 beta units. So, mainly what is the message here? The blood of the fetus is made up of 4 subunits, out of which 2 are alpha and 2 are gamma. What are they? 2 are alpha and 2 are gamma. Now, 2 interesting things happen. Whereas the mother has more DPG, a pregnant mother has more DPG, which means what? Which means what? She will give away the oxygen more readily. Okay, on one hand. On the other hand, the fetus will have alpha and gamma, and alpha and gamma combination has almost no affinity for DPG, which means it has more affinity for oxygen. Are you with me? If what will the DPG do? Give away oxygen. So, if you have no DPG, what do? You bind more oxygen. Are you with me? Yes or no? Look at yet another trick of the hemoglobin molecule. So, the fetus, by having in its blood hemoglobin that is made up of 2 alpha units and 2 gamma units, it has DPG, but DPG cannot combine, okay? Which means it has more affinity for oxygen, which means the mother, okay, in her body has an additional part which we know is a fetus, which has far more affinity for oxygen. The oxygen will directly go before it goes anywhere else from the blood of the mother into the blood of the fetus. Do you appreciate the point? Okay, why the nature has come up with this amazing idea that in the baby don't give 2 alpha 2 beta, give 2 alpha 2 gamma, okay, which has less affinity for it, it doesn't combine with DPG, more affinity, it can draw more blood, but eventually after the birth the baby will lose the gamma unit and the baby and you will have a combination of 2 alpha and 2 beta. Here we have the green one is our classical here, this is our classical 2 alpha hemoglobin with 2 alpha 2 beta units, and the rest of the story is same, 20 this everything is same. But as a result of the this is adult hemoglobin adult, this is HbF, F stands for fetus or fetal. Okay. As a result of that, can you clearly see for at this partial pressure, whereas it can have so much of saturation, at the same partial pressure this have so much saturation, means it can combine far more oxygen, means affinity for the affinity that is shown by the hemoglobin of the fetus is far more than that of the affinity of the mother. So the mother's blood will take oxygen at the level of the lungs, it will go everywhere. As it goes into the placenta, on the other side of the placenta there is baby's blood. That baby's blood has hemoglobin alpha alpha gamma gamma, it has far more affinity. So, the baby will ensure that it will keep on drawing more oxygen which is required for its own development. Okay. All right, we will stop now. There are some videos I have given at the end, please do, I am at the end of this talk.